Here we will look at organic compounds, carbohydrates, and lipids for this next review of chemistry. We will begin by looking at carbon, which plays a fundamental role in organic chemistry and forming organic compounds within the body. So to recall, carbon is the element of C, and so if we look here, carbon has 6 protons and it has 6 neutrons, okay? And then it has 2 different valence shells, all right? It has the first one, which has 2 electrons, and then the second one, which wants to hold 8, but since it only has typically 6 electrons, it only has 4 in that second valence shell. So what that means is that it can form up to 4 bonds, okay? So when we have 4 different bonds, here's an example. Here it has a bond with a hydrogen, another hydrogen, and a third hydrogen, and then this last bond then is with another carbon. And so here what we have is the molecular formula for ethane, C2, because we have 2 carbons, and then we'll have 6 H's because we'll see what is attached within this molecule. Carbon also can form a double bond, and this is where we have 2 pairs of electrons are going to be shared. And so that's represented here in this figure, okay? And here we have the double bond where those 2 lines represent a double bond, okay? So we'll have same number of carbons, so 2 carbons. And then now though, the difference is only 4 hydrogen bonds because 2 of those electrons are being shared between these 2 carbon atoms. Hydrocarbons are going to be molecules of only hydrogen and of carbon. So these are important because when we break these bonds, they release lots of energy. So when we undergo digestion, this is when we have our form of energy that are being required. All right, so we have different types of hydrocarbon structures that our body can use. So here at A, what we have is a carbon chain, and these help to produce different types of backbones for other functional groups, which we'll talk about next. The second type that we have are going to be these different types of rings and have a similar purpose where they can add different types of groups onto these molecules. And these are all arranged and only are single bonded to each other. The third group that we have here in C is a double bond ring structure, which too can add different functional groups. And then what this allows for are organic compounds to be classified into groups, which then have unique chemical properties. So some of the functional groups that we'll see again and again include carbonyl groups. And so this is where we have a C double bonded to an O somewhere within the hydrocarbon structure. We'll have a hydroxyl group, so a lot of our alcohols. We'll have sulfhydryl groups, where it's a sulfur and a hydrogen. We'll have amino groups, which we'll see a lot within proteins and then also within DNA, and then hydroxyl or carboxyl groups with the COOH, and so this forms a weak acid. Phosphate groups, which adds a phosphate, and this is important for a lot of cellular recognition as well as protein recognition as well. Then we have different types of ketones, and so these are a lot of breakdown products of triglycerides. Also we'll have organic acids, which are the same thing as carboxyl groups. Aldehydes, which can actually reduce to form and make alcohols, and then we saw alcohols already with the hydroxyl group. With organic compounds, we can also have stereoisomers. And so these will be molecules that have exactly the same atom arranged in exactly the same sequence, but differ in spatial organization of their functional group. And this is going to be critical to its function. So a given enzyme may interact with one isomer but not with another. This also comes into play with pharmacological agents that have been, or pharmaceutical agents that have been on the market where one isomer, stereoisomer, will not penetrate, say, the blood-brain barrier, like in the terms of a lot of our neurodegenerative diseases, because they're not biologically active then. And so when we look at this example, what we have is a difference here with the cis versus trans. And so this just has to do again with spatial orientation, where really what I want you to focus in on is here we have this carbonyl group, and instead of being located up at the top left-hand side of this carbon, now it's been replaced on the bottom left. We'll see this time and time again that occurs naturally within sugars. Glucose, galactose, and fructose all have the same number of hydrogen and number of carbons and oxygens. However, their arrangement, again, is just a little bit different. So here's an example of glyceraldehyde, another form, where here we have this alcohol group has just switched sides. And so sometimes this does not affect, again, may not affect the body, and so may be absorbed just easily throughout the bloodstream. But there are times when a different isomer means that the body will not absorb it, and it will just be excreted within our urinary tract. Carbohydrates are a form of an organic molecule that contain carbon, hydrogen, and oxygen. And they always have the same ratio of 1 carbon to 2 hydrogen to 1 oxygen. So the formula that we come up with is CN, H2N, and ON, just N representing whatever number. So for example, with glucose, we have C6H12O6 representing that carbohydrate. So our carbohydrates serve as a major source of energy within the body. It includes different sugars and starches, which some starches we cannot break down, such as fiber, but also some sugars that just will pass through the bloodstream and will affect us differently. Typically with carbohydrates, we always see an "ose" ending, and so this usually denotes some type of sugar. There are 3 main classifications of carbohydrates. We'll start from simplest and go to more complex. The first is monosaccharide, and so mono meaning one, saccharide meaning sugar. So the definition is a simple sugar and it has 1 carbon ring. Examples of this include glucose, which is what our bodies typically break down and use for energy. Another example is fructose, and fructose is what we typically find within fruit, and then galactose, which is found in dairy products, not as sweet as glucose though. All of which of these will be stereoisomers to each other, but they have the same formula, C6H12O6. The next complex structure or classification of carbohydrate is a disaccharide, and di meaning 2. And so here we have 2 monosaccharides are joined together by some type of covalent bond. And so examples of this are sucrose, which is commonly known as table sugar, lactose, which is galactose and glucose. We find this within most dairy products, so a lot of people who are lactose intolerant this is the molecule that they cannot absorb. And then also we have maltose, which is not as common, but we do find this like, for example, in malted barley, which we find within beer. The third classification of carbohydrates are polysaccharides, and these are made up by several different monosaccharides that are joined together. And so common examples that we have are starch, glycogen, and cellulose. Starch is typically what we find within plants, and so this is how they store sugar that we can break some of these down and then use for to make into glucose molecules. All right, and so here in this figure, so here'd be one glucose molecule plus another, and so this can continue with a different chain. Glycogen, we use this to store sugar in animal cells, and so it doesn't pull apart in water, and like simple sugars do, so this is for more long-term energy use. And then cellulose makes up plant cell walls, however, we can't digest that, and so typically this is what we associate fiber with. One of the reactions that occurs within our body is called dehydration synthesis. And this is where we use for fats, proteins, and nucleic acids, not just for carbohydrates. But let's just go through carbohydrates here to explain the process. So with this, what we have are, in this top panel here, we have two glucose molecules, and what they will do is form one molecule of water and a 2 sugar chain, and so this will make maltose. So with this, what we have are covalent bonds that hold monosaccharides together, and so these are going to be formed by dehydration synthesis, where the hydrogen atom from one molecule and the hydroxyl group from another molecule help to make water. So here in panel B, here's another example. So here we have our hydroxyl group, and then here one hydrogen group. And so those are going to unite and form then what we have as sucrose. So we go from monosaccharides to disaccharides. And so this can continue on until we have a polysaccharide chain. Another reaction that occurs within the body is hydrolysis. And so this will be the opposite, where with dehydration synthesis, we're creating bonds, but now with hydrolysis, we are breaking bonds. And so this is between saccharides, and so now we're adding water to split the molecule. And we use this also to break down different fats, proteins, nucleic acids, as well as carbohydrates. So here we have an example in panel A. Here we have starch. And so here's a long chain of a polysaccharide, and then we're adding water now into the chain. And so what has created then are forms of maltose. And so we go from a polysaccharide to a disaccharide, and then further we can continue this process where we add more water, and so we can take this maltose, which again is a disaccharide, add water to it, and now we're making molecules of monosaccharides, which then is important because then our body will use that as energy that can be converted and rapidly broken down to be used. Now let's move on to lipids, and so we'll start with triglycerides or triacylglycerols. You'll see those terms interchangeably. And so lipids are composed of nonpolar hydrocarbons, so they're made of hydrogens and carbons, as we learned earlier, chains as well as rings that makes them hydrophobic. And so they're hydrophobic, meaning that they're insoluble in water because they're water-fearing. So this now includes different fats, which are in solid states, and then oils, which are in liquid states. Triglycerides can be broken down into two different categories. We have the first, saturated fatty acid, and then unsaturated fatty acid. So first of all, fatty acid, that just represents a long, nonpolar hydrocarbon chain. So here in this example, very long with just hydrogen and carbons. And then at the end here, what we have is this carbonyl group, okay? And so if every carbon, like in this example, what we have, is bound to only a single pair of hydrogen or to another carbon, making only a single covalent bond between all of its atoms, then this is termed to be saturated, okay? So this means that all the carbons are fully saturated with the maximum amount of electrons that they can have. An unsaturated fatty acid, like in this example, here we have now where we have double bonds and between the carbons. So they still have a long hydrocarbon chain, in the carbonyl group, but now instead of having single covalent bonds, what we have are double covalent bonds. And so that's going to be the difference between now saturated versus now this is unsaturated with the amount of bonds that carbon can have. Under certain conditions, our body has this mechanism to produce glucose from non-carbohydrate sources, and this is through ketone bodies. So with ketone bodies, what happens is that triglycerides undergo hydrolysis or breaking of bonds, and then it forms these free fatty acids in the blood. Then that can be used for energy or can be converted by the liver. And so here in this example, acetoacetic acid can be converted into glucose, but then one of the byproducts is loss of carbon dioxide. And so people who undergo low carb diets, undergo starvation, and have uncontrollable diabetes, what they have then is this excess production of acetone, which tends to give that fruity smell to breath. And so over the long term, though, these are not ideal because you start excreting this within the urine, and this lowers the pH. And if these are high enough, then what happens is that can lead to coma or death. Because these tend to increase the amount of hydrogen ions that are circulating within the bloodstream. Phospholipids are very important for cellular membrane and then also for cellular communication. And so phospholipids, these are going to be lipids that have a phosphate group and a major, again, component of the double layer within our cell membrane. These molecules are polar, and they also have a hydrophilic phosphate that's going to point outwards on each side. And so here in this picture, here is our hydrophilic, which water-loving side, and then it will have a hydrophobic fatty acid and glycerol chain that's going to point inward. So if we looked on the opposite side, then it would look something like this. And so here now we are creating that double membrane. So here is the inside of that membrane. And so say this phosphate head on the left, this is going to point inside the cell. And then this opposite head right here is going to be exposed to the outside of the cell. And so both of which, here and here, these will be water, or will have a lot of water with them. And so that's why these hydrophilic portions with the phosphates are going to be towards that water region. Another type of lipid are called micelles, and here we have an image of them, and these are going to be polar and help to decrease the surface tension of water. We see this, and we'll talk more about this when we get into the digestive system, where these micelles will form, and what that helps to do then is to help increase the surface area to volume ratio so that we can have digestion of these fatty acids so that we can use those for energy. We also find micelles found within the lungs, and so surfactant will have them. And so the purpose of these micelles then is to prevent our lungs from collapsing on each other, especially when we talk about those smaller alveoli. Steroids also fall into the category of triglyceride, even though they are structurally very different. However, since they're nonpolar, they're considered to be a lipid. And steroids are very important for the formation of different types of hormones. And there's many different types of steroids, but all of which have the same feature. They have a 3,6 carbon ring, a 1,5 carbon ring, and then some type of functional group. And so that functional group will make up different steroids. So here, if we look at cholesterol up here, so here we have our 3,6 carbon ring. Here's our 1,5 carbon ring, and then this would be the functional group. So cholesterol is going to be the precursor for making all steroids, And so testosterone, estrogen, and corticosteroids would be an example, but for the development of these hormones. And then also we'll have cholesterol used as a steroid to make different molecules like vitamin D and then bile salts that are produced within the liver.